In this screencast video lecture, we are going to see about the importance of ATP and its generation. That is how ATP is generated there in the living systems. ATP refers to adenosine triphosphate. It is the primary energy carrier of the cell. Often it is referred as a energy currency of the cell, since it transfers energy for many biochemical cellular processes that have been operating in the cells. Cells use their supply of ATP in order to power various energy requiring processes. For example, to make sugars, to supply activation energy for various biochemical reactions that have been taking place in the cell. ATP is basically a nucleotide group, that is, it consists of three different components. That is an adenine, which is basically of a nitrogenous base, and the type of sugar present here is a ribose, which is a 5-carbon sugar, and it contains three phosphate groups, which are the sources of the high energy bonds that have been present there. Especially the last two phosphate bonds are the one that are carrying the high energy bonds. So on breaking of the particular bond, a lot of energy could be released there in the cell. So that is the one that have been referred here in the name of a ATP cycle. It means how ATP is being cycled there. ATP on hydrolysis, it will be giving into the adenosine diphosphate and inorganic phosphate and energy. Alternatively, in the ATP cycle, the energy that is required for conversion of ADP to ATP is obtained there from the respiration or photosynthesis. Now we look at the important functions of the ATP there in the cell. ATP provides an immediate source of energy for a wide variety of cellular reactions. The first one is the various kind of biosynthesis reactions. Basically they are of anabolic reactions, that is synthesis reaction. ATP is required there as a energy for formation of proteins from amino acid, formation of DNA or RNA from the nucleotides, synthesis of lipids and carbohydrate from their monomeric substrates. Next one, ATP is required there for the muscle contraction. Especially in the eukaryotic cells, ATP powers the interaction between the actin and myosin proteins in the muscle cells, which enables a proper movement of the muscle fibers. ATP is the primary energy source for the transport system that have been operating there in the living organism. It is basically required for the transport of molecule against the concentration gradient, which is technically referred as the active transport system. Here it is required for various transport proteins or transport pumps, which are referred as a sodium potassium transport protein or sodium potassium ATPase pumps, proton pumps and calcium pumps. All these require energy in the form of ATP in order to accomplish their transport process. Next one, ATP is required for the cell signaling. Mainly it is involved in the signal transduction process that takes place commonly in between the cells there in the eukaryotic organisms. Mainly it serves as a second messenger, that is cyclic AMP, adenosine monophosphate, that have been derived from the ATP, is serving as a second messenger there in the signal transduction process of the cells. The next one is the role of ATP there in the exocytosis as well as endocytosis, some special kind of a transport process that have been taking place there in the eukaryotic cells alone. Here ATP provides the energy there for the vesicle transports. Vesicles are bubble-like structures that have been present inside the cell. They are made up of lipid layer and they involved in the transport of various synthesized molecules within or between the cells. Say for example, they involve there in the release of neurotransmitters, hormone secretions inside the cells, and they are essentially required for the process of phagocytosis and pinocytosis that have been involved in the eukaryotic organisms. Next one, we look at the role of ATP there in the DNA and RNA synthesis. ATP is one of the building blocks of the RNA, that is of a adenine nucleotide, and it is essentially required there for the process of replication as well as transcription that have been taking place there in the cells. Next, we look at the important types of catabolic pathways that have been operating there in the living organisms and the type of phosphorylation that have been involved there for the synthesis of ATP. The first column shows the pathway, that is glycolysis is a pathway in which ATP is produced by substrate level phosphorylation. Glycolysis is commonly operating there in the cytoplasm of the cells. The ATP yield during this process is around 2 ATP. Whereas if you look at into the next one, fermentation, which has been operating only under the anaerobic condition and in anaerobic organisms. Say for example, yeast shows fermentation, Clostridium shows fermentation. There, the ATP is again obtained by substrate level phosphorylation. The location of this process is there in the cytoplasm. Again, a 2 to 3 ATP yield could be obtained by this process. The next one is a Krebs cycle, which is a follow-up cycle of the glycolysis. Here also in a step, ATP is produced by substrate level phosphorylation. It commonly takes place there in the mitochondrial matrix or sometime there in the cytoplasm of the prokaryotic cells. Here the quantum of ATP generated is only 2 ATP, especially by the substrate level phosphorylation. Then we look at the next one, electron transport chain, which is the one operating there in the inner mitochondrial membrane of the eukaryotic organism, or it may be operating there in the cell membrane of the prokaryotes. Here the ATP yield is 34 ATP. The type of ATP that have been generated there in the electron transport chain could be classified into two groups. One is an oxidative phosphorylation, the another one is a photophosphorylation. Next we look at into the amount of ATP produced there in the prokaryotes as well as eukaryote. Prokaryote generally produce about a 38 ATP per molecule of glucose oxidized there in the cell, whereas eukaryote is producing a little bit lesser than that of the prokaryote, that is 36 ATP is produced per molecule of glucose oxidized there in the eukaryotic cells. Next we look at in detail about the different types of ATP generation mechanisms that have been operating there in the cells. ATP can be generated by three different ways in the living organism. It is based on the needs, environment in which they grow and the type of the substrate that have been used for their growth. These are the things that governs the type of ATP generation there. The first one is a substrate level phosphorylation. The substrate level phosphorylation refers to direct transfer of inorganic phosphate group from a high energy substrate molecule. For example, here in the glycolysis process, you can look at here the two steps in which the ATP has been generated. Here the high energy phosphate carriers are phosphoglycerate as well as the phosphoenol pyruvate that donates their phosphate in order to convert ADP to ATP, and this particular phosphorylation is referred as a substrate level phosphorylation. That is, phosphate addition have been taking place just on the substrate level. There is no any electron transport chain has been involved there in the ATP production. Apart from glycolysis, this substrate level phosphorylation is noticed there in one step of the TCA cycle or Krebs cycle or citric acid cycle. In addition, the ATP generated through the fermentation process is also operating at a substrate level phosphorylation. The next ATP generation type or mechanism is the oxidative phosphorylation. Oxidative phosphorylation refers to ATP synthesis that are driven by the flow of electrons in an electron transport chain, mainly in the presence of oxygen. When oxygen is present, it is referred technically as aerobic respiration. Instead of oxygen, any oxidized form of inorganic compound such as a nitrate, sulfate are present, they are referred as an anaerobic respiration. Here, energy source for the phosphorylation is obtained by transfer of electron through certain electron carrying molecules such as NADH and FADH. These molecules are all originating there in the operation of a glycolysis and a TCA cycle. So these energy carriers are all channeled there into the electron transport chain, which donates their electron and proton, that leads to the formation of a proton gradient. A proton gradient is established across the membrane by hemiosmosis mechanism, and subsequently the proton is trying to flow back into the cells with the help of some special proton pumps which are technically referred as a ATPs. So by flowing back of the protons into the cell, aided through ATPase, the phosphorylation is effected. That is, ATPase pump is the one which effects the phosphorylation of a ADP with inorganic phosphate, resulting in the formation of energy in the form of ATP. The next mechanism is a photophosphorylation. It is a process of ATP synthesis that have been driven by the energy obtained from sunlight, that is through absorption of sunlight during the photosynthesis process. It commonly occurs in the thylakoid membrane of the chloroplast of the plant cells and also in certain photosynthetic bacteria like cyanobacteria, where the thylakoids have been housed there in the cytoplasm. Apart from that, they have been taking place there in certain anaerobic anoxygenic group of photosynthetic bacteria such as green sulfur bacteria. In this process, during photosynthesis, the light energy is used to derive the electron transport chain that results in the generation of a high energy electrons and creation of a proton gradient. At the end of the electron transport chain operating, the flow of proton backs into the membrane with the help of again the special kind of a ATPase pump, which leads to the synthesis of a ATP molecule. That is, this particular ATPase pump is the one that helps to phosphorylate the ADP molecule into ATP, and the type of phosphorylation is referred as a photophosphorylation.